the concept of physical samples. That might be things like data management of like the metadata or data associated with samples. It could be the actual management of the physical samples itself, like the storage areas, the curation processes, the reboxing, and also some of the, as you think outward about physical samples, sharing and access. Again, that connects back to the metadata and the structure you might be creating around the samples, but really connecting out with the users and what the workflows might be in order to engage them in different areas. My co-chairs for this session are Val Stanley, who's online, and Andrea Thammer, who's here in the room. And to go over the agenda, today we're going to spend some time with just some news and updates from the community, and then we're going to go into community reports. So those of you in the room, if anyone wants to share a project you're working on or a related topic that you just want to share here in the room, we'll have some time for that. We have some people already volunteered, but we've given altogether with this news and the reports about half an hour, so we have time for those. After that, we're going to work on the major activity that this cluster's been working on is a set of sample sharing guidelines. And I know every time breakout, people want to like sneak out of the room. I get it. But hopefully for this, it's a lighter weight thing. We're going to be looking at some Word documents. We already have someone fleeing when they heard breakout activity. It's okay. We'll be working on some Word documents. You can also do that by yourself. We won't make you engage with others. But we will come back from that to talk about some of the activity work that you've done. And then we'll spend some time just talking about next steps and wrapping up the session. Okay. So anyone have any news or updates to share? Normally we often have someone announcing a new project or something, but I've got something, but I'll pause a moment to see if anyone else has anything you want to share. Shelley? Yeah, we have more online people.
Sarah, I think you were aware, and maybe a couple folks in the room are, but we're working on this concept around a collection DOI for, especially when you're citing, the original use case was citing over 100 data sets. But we realized that, of course, there's other things that could be cited. So that would be of interest to the group. It's the charter is about to be submitted to RDA to start a working group within awesome. The ESESIG, and it would be very relevant to citing samples.
I think that would be useful because that's a challenge when we've been working on this guidelines, and we'll get into that a bit as I said in the breakout of how do you cite 1,000 samples in a paper. So I thank you for bringing that up. I think that's really relevant. Andrew, do you want to?
I just had a question. Is that like collecting event DOIs or like a data collection or natural history collection?
So we had a vocabulary problem during the project. Collection meant something, crate meant something, package meant something. So we actually named the thing a reliquary. It's a humorous thing. It's not meant to stick, although it's sticking. And it is funny though, isn't it? So we didn't, it's in definition. It's not using any other definition. And maybe it will eventually, but at the moment, so it's, so imagine a list of DOIs where you get primary credit for that list and you hold it with a collection DOI. That's a data site term, by the way. You hold it with an umbrella DOI, whatever the name will end up being, and then it makes sense somehow. And creating it, unpacking it, making sure that the authors of each of the children, each of the elements within the reliquary, whatever we'll call it, are acknowledged, and I'm avoiding the word credit because I'm seeing Mark, there's an opportunity for there to be acknowledgement that the use was made. And we did rewrite our charter because of Mark's feedback on the word credit.
Say if you have an opportunity, Shelley, it'd be great if you can add if there's any links to that RDA group, to the meeting notes. That way people can see it to join. RDA is really easy to join. Just sort of have to create a login basically to their website, and you can engage in those communities. Any other news? from the audience or online? Do we have anyone online who's got any news items they'd like to share?
Just to plug our session later this afternoon.
Oh, good. I was hoping
the
you'd do that.
Research Artifact Citation Cluster we recently published a paper in EOS talking about how to credit various research artifacts. So not just data, but software and semantics and learning objects and all those sorts of things. And so that was sort of a milestone for us. And now we're kind of figuring out where to go next in terms of how to, I think, really focusing on the data aspect and the different roles involved with that. So come join us this afternoon.
Thanks. Yeah, and congratulations, you guys. Right. And one last news item then too is, so Leslie Wyburn and I, are you going to talk about our AGU session?
No, I'm
Okay.
going to add on to Mark. I'd like to say that we'd love to get something done on credit for samples, because one of the difficulties with samples is that people go and slog themselves to death and work extremely hard to go and collect the physical samples, but once they're in the systems, a lot of data is generated by people using those samples. And one of the things that's so difficult if you're trying to preserve samples is that you've lost your credit, because people don't get back to crediting the person who collected the sample, who's often quite separate from the person who's doing all the analytical work.
Right. So no, I was just going to, I mean, I think we're just going to keep reinforcing ourselves, each other. But so samples was one of the things that we looked at in terms of trying to figure out how to credit different things. And one of the things that emerged, and Sarah led that sort of subgroup, was that samples are a whole different critter. And so I think that's why this group formed, right? I mean, so that, but maybe not. So maybe that's an issue, because yes, we're trying to figure out what's next. So whether we bore into samples or different data types, one of the conclusions that we realized is that different data types have different credit schemes. So like field data collected in the field, data collected from a satellite, data collected from a sample, all have different roles that are involved that deserve different levels of credit depending on the context. So yeah, we might want to tease that out further.
Yeah, the main thing I think it felt like is that credit group is working towards data as a focus moving forward. And so we were needing a new home for the concept of, well, what about credit for samples? And yeah, exactly. But Mark was just saying our samples data, and if we go back, I used to say they were, then I got told they were not, and now maybe they are again. So we'll see. That made me think of something else. Now it fell out of my brain. Sorry, I'll come back to it if I remember it. But that will tie into what we're doing in the breakout activity. We're looking at these sample sharing guidelines and any recommendations for, well, how do you cite the person who collected the samples? Where does that live in the citation? Where does it live in your document, your published work? So that these things will hopefully get addressed tangentially in our activity today.
Sarah, we have a raised hand in the Zoom.
Yeah, I'm sorry, I can't tell what your name is. If you want to go ahead and speak.
Hi, my name is Diana. I'm sorry, I tried to change my name on the thing. I can't figure out. Sometimes I can do it, sometimes I can't. Hi, so I am, I'm totally new to ESIP, and but I'm very much interested in this issue in sort of the general realm. I'm primarily interested in data, in providing attribution for data sets. But one of the issues that I wanted to bring to your attention, I think it's not maybe so relevant for earth sciences, but if you want to connect with biomedical data, for instance, you also need to consider the possibility that how do you cite if the subject is the data? For instance, if it's your DNA, or if it's your blood, or if it's your social information, that's something that the biomedical realm is grappling with. And you know, if it's your medical information, do you have any attribution, and do you have any control over that? So
Yeah, that's a very interesting perspective. You should come to the credit session this afternoon.
I'm coming.
right.
I'm absolutely coming. Yes, this is a very relevant issue. So yeah. But yeah. I'm fascinated, and I really want to understand all of the issues that Earth Sciences faces. Thank you.
Yeah, thank you. Yeah, I feel like that's a perspective we haven't even touched on in that group. So yeah, please bring that up again this afternoon.
Absolutely.
And Mark says it kind of scares him, so I'll just share that with those of you online.
It should be scary.
And so one final bit of news items is that AGU abstracts are due August 3rd, and we actually have a session dedicated to samples. So now that I see who here is interested in samples, expect to be browbeaten to submit an abstract to the session at some point. If you add your name to the notes, maybe I'll email you all to remind you about this. You can get to the notes using, oh, sorry, there's a bit.ly on the screen, but it's blocked by all the Zoom stuff. Actually, if you can see those last digits on the end, it is just bit.ly/ and then those characters you can actually see on the screen. So you should be able to get to it. Hopefully, it's the auto transcripts are covering it, sorry. But yeah, there's a session on samples, and we'd love to see abstracts to it so the session can make for one, right? You got to get enough abstracts. But we've got some really lovely invited talks from some early career folks, and it'd be great to just engage more people in this discussion about samples. Not here, we've been very welcomed in ESIP. It'd be good to continue that conversation too in other communities like AGU. Yeah. Abigail?
Thanks. I just wanted to also share, this is Abby Benson. I'm chair of the Biological Data Standards Cluster. So that cluster, we have a primer for standards for biological data, just so you all know about that. And in that cluster, we've been trying to host sessions on panels for the different sections of the primer. But the one thing I wanted to bring up for this group is that the Biodiversity Information Standards, also known as TADWIG, does have a connection here in that we're trying to reuse the iSamples work for defining a material sample for in this biodiversity realm. So we're trying to make that connection there. And so I just wanted you all here to know that that's happening.
And I believe they're organizing a workshop on that, because I can't remember her last name, but Teresa, I'd been emailing with her.
Yes, the workshop happened yesterday, so I couldn't attend sadly, but I hope to find out more about that workshop, next time we meet.
Yeah, because I'd asked her so they're going to host a session in TADWIG to then review these metadata standards at the iSamples group, which we've talked about here in previous meetings, but I can clue anyone in on what that is later. But has created these meta vocabularies of what are a sampled feature, a sample type, or a material type that might be going across domains. So I'm going to one central catalog to look for samples, and I might use certain terminology and then be able to discover samples in both biology, earth science, archaeology. And so they're road showing some of these vocabularies in different places, and this workshop might be one way to see, does this vocabulary work for the biodiversity community? And it would be great, the organizers have said it'd be great to get people from the earth science to come in as well as and engage in part of that. So yeah, that's a good point. When we get more information, we could definitely share it with this community. Oh, Leslie's got more news. Good thing we build in half an hour for, discussion.
So in Australia for research infrastructure, every 5 years they do a roadmap. And the roadmap was just released in November. And it actually has a section saying physical collections are a vital resource for research and underpin activities that range from health and medical research to ecology and agriculture. Physical collections are critical to support blah, blah, blah. And the important thing is, once you get physical samples into this research infrastructure roadmap, it then opens the door for you to be able to put funding bids with various funding agencies against it. So we thought it was an incredible achievement because these have been going on since about 2008, and finally we got physical samples into it.
I think that's a good point, is that even when we think about what clusters do and the activities, it's great that we have this sample guidelines that we're working towards, but even just creating awareness for our domain and our area and getting it worked into the system and making those connections is a really great outcome. So glad to have many of you here, and hopefully you can carry this back into your community as well. Okay, so let's move on to there we go. We've got some community reports, some folks, and we might have ended up, we might not take this full 25 minutes because I think we're already, I've lost the clock, but I think we're already at about 20 minutes into the session. Let's see. But Leslie, did you want to share updates from the Down Under? I think that you've got two on here, this one.
Okay, so as I said, it's a big achievement to get something like that in an infrastructure roadmap for the whole country for research. And the Australian Research Data Commons is now going to form a community of practice for information management for physical samples. And I don't know whether you know, there's actually a group called E2SIP, which tries to coordinate equivalent activities in Australia with here. And we did this fairly successfully with the the information quality group that ESIP has, and we had a parallel one in Australia. It's just the time zones kill us. Otherwise, we'd just join you guys. So, like, it's 11 o'clock at night back home. So we will run, but what we would very much like to do is keep in parallel with what you're doing, particularly this work on accreditation in journal publications. And if something happens in the credit cluster, we'll be trying to do parallel, which means we might be inviting some of you to speak at our sessions, but we promise we'll try and make it at a reasonable time for you.
Okay. And then Andrea, did you want to provide an update from Spinach? Do you want to provide context, or do you want me to?
I can provide
Okay.
context. Yeah, hi everybody. I'm Andrea Thomer, and I am starting at the University of Arizona. I know some of you already, and I'm moving, if you don't know that, from Michigan to Arizona. So this year we presented, there were two presentations related to this cluster at Spinach. So for those of you that might not, has anybody heard of Spinach before? It's my favorite acronym for a scientific community. It stands for the Society for the Preservation of Natural History Collections. So it's a lot of natural history museum collection managers, curatorial staff, sometimes registrars. So really people that are doing a lot of the nuts and bolts of caring for physical collections, but increasingly over the last 10 to probably, I'm pausing because, so like a lot of these folks I know were like early adopters of database technology, and they built a lot of the really fundamental infrastructure that makes it possible to share data about samples. But over the last like 20 years, they've been really thinking about digital specimens, so what it means to share information about physical samples on the web. The focus tends to be on biological collections, but there's also some interest in earth science collections there. So we had a poster talking about our sample citation guideline efforts there, basically outlining what we're doing in the cluster and trying to advertise, look for collaborators basically, because those collections have a lot of similar issues with getting citations for the usage of their specimens, as we see with the ESIP related sorts of collections. And I also presented on some work with Sarah Lafia, who's a community fellow here, where we're trying to extract biodiversity specimen citation from biodiversity literature, and then therefore show the impact of those collections on the scientific literature over time. The long and short of the impact from this is that like both of these presentations were pretty well received, especially because I wound up having to present them virtually, which is good because everyone at Spinach got COVID. Yeah, but the biggest takeaway I think is that there is a lot of energy there for trying to build citation guidelines together, or at least keep in touch about specimen citation guidelines between the two communities. One person that reached out to me after my talk is named Donat Agosti, A-G-O-S-T-I, who is the head of Plotzy.org, which is been building tools to mine biodiversity literature and taxonomic treatments for specimen citations for a while. And he's also involved with Pensoft Publishers, which is a set of journals for biodiversity and taxonomic literature. And so he's very interested in keeping in touch and trying to build specimen citation guidelines that, maybe if they're not exactly the same between the communities, are at least in conversation with each other, because there are going to be different needs of both of the communities. I think that's the main update. It was really gratifying to see that folks were interested in this there. And it was both validating and frustrating that they have the same problems. And so there's just the journals in that universe, they don't have specimen citation guidelines either. So there's a lot of work to be done, but at least there's people that are interested in doing that work.
Yeah, and it's great to see. I should have put the poster up, but I'm not sure how to find it.
Oh, I put a link to it in the notes, as well as my and Sarah's slide deck from our project. If you scroll down to the spinach.
Is it touch screen? No, sorry, the computer is very weird. I can't. I'm stuck in the table, sorry. There we go.
And thanks to, so Sarah took the lead on this poster, and there were some awesome 11th hour assists from various cluster members. So thanks to those of you that did that.
Here, I'll make it full screen so it's easier to see. Clearly, we don't expect you to read that from back here, but this was at Spinach. I guess I didn't think about that. We could have put it up here. Where were you last week, Mark? But I think it's really great that the purpose was to make this connection with these domains, and then we did, right? Like, so I think that's a resounding success for the cluster. So thank you all for all your work on it. And then also, especially to see that now we'll talk about this later, we were like, what are the next steps once we get the guidelines done? Looking for reviewers, looking for feedback from other communities, and finding other adopters. And so we're starting to make some of those metrics happen. So that's great. And then, did you want me to share Sarah's poster?
It's a slide deck.
Oh, is it? She's not here.
Yeah, we can come back to it.
Okay, if she shows
comes
up,
in.
we'll
Yeah.
give her some time. Okay. Okay, but thank you, Andrea.
Awesome.
Okay. For those of you who are not aware, there is a physical samples interest group within the Research Data Alliance, RDA. They've met in, sorry, my brain is melting, early June? Is that when RDA happened this year? It was part of International Data Week. So just wanted to give space just to remind people that this group exists and to encourage them to engage there as well, because again, a lot of these communities provide opportunities to either talk to other domains or also to reach different audiences. RDA is a very international group and community, and so it's a great place there too to learn what other people are doing in other areas. They had been doing a webinar series that we were doing as a joint ESIP RDA webinar series on physical samples. We paused that in December just because of the holidays. It got a little hectic, and then we haven't picked it up again. But if there's anyone who's interested in that as a concept, either volunteering to give a webinar or perhaps even volunteering to help run that series, it was very well received, and there are some of the talks are on the RDA YouTube channel, and you can view them there. I believe they're also looking for a new co-chair. So if anyone is already engaged in the RDA section and wants to do more and connect better, that is also an option as well. The current chairs, Leslie is one of them, along with Kirsten Leonard and Jens Klump. And what is Esther's Plum? Yeah, but Esther would like to cycle off, I believe. And so they're looking for another co-chair. Abby, did you want to talk about the biodiversity standards anymore, that cluster? Yeah, that's why I was like, oh, I forgot to tell you, I marked you in the slides. Sorry about that. Okay. Okay, good. Oh, good. Maybe that will get us back a bit on time. Perfect, actually. Oh, wow. Just before we move into the breakout activity, does anyone else have any other news or ideas they want to share that maybe they thought about in that, since we actually do have 2 minutes? Anyone online, perhaps? Okay, all right, then we'll move on. I'm just shocked. How do we get back to being on time? Okay, so today we're going to focus on, we've mentioned this briefly, but we'll get into a little bit more, our sample sharing guidelines. And I'm going to give a brief overview of the project, and then we're going to work in breakout groups for about 30 minutes. We'll come back and do some reporting from those groups, and then turn that into a conversation. Well, what do we do next? And then we'll close the session with just some wrap up and general points. All right. So what were the motivations for this? So as a cluster, we got together and we're like, all right, what are we going to do as an activity? Not that clusters have to have an activity, but it's good to have some sort of thing that we can be working towards. And a common conversation point, and I think part of it was coming out of that credit cluster as well, was, okay, samples should be cited, but how do you do that? And we need more information and recommendations, because it'd be really great if we could, as a community, come up with a shared understanding of how to do that process. And so we started working on it by ourselves on a document, and we were presenting it on one of the clusters sessions, I believe it was, when Shelley and Chris made some connections to, well, AGU would also be interested in some citation documentation for samples. In particular, they've been working on the data and software guidelines for authors. And so asked us to create something similar for samples. And it was a great opportunity because, as I mentioned, we've sort of been working on this in a nebulous state. But the data and software for authors gave us sort of a template and a guideline of these are the sort of topics we should address. Here's how some other group has already talked about these concepts. And so it was great to use that as a base. And so what we've done is we've taken these data and software guidelines for authors, made a copy of them, and then we're just changing everything to say sample. And where it's not related to sample, we're saying, well, what is related to sample? And so as a cluster, that's what we've been working on for about, I want to say the past 6 months, is just going through this document and saying, well, where are there sections that are missing? Is this topic relevant to samples? And with the ultimate goal of transforming it into a document that is then related to the needs for authors for samples. And to give a little bit more about the tone, the idea is that this would be shared by AGU as the publishers with providing guidelines and information to authors who are submitting papers. So in this point, we've also narrowed the tone and the conversation down as well to that specific viewpoint. And perhaps once this is done, we might move and think about other perspectives, but it was good because it gave us a very actionable thing to do. And so the current status is we're reviewing content of the document, and you can see a public version from the bit.ly here. You should be able to comment on that document. We also have a cluster-specific version, but I just thought it would be easier to share this public version we created because it's got a lot of things and notes in red that sort of give context to what we've been doing. But there's some sections that we've already worked on as groups that have been refined, and then there's some sections that we haven't touched at all. And that's what we want to focus on in the breakout activity today. Is we have a few sections of this document that were, again, borrowed from the data and software guidelines that we want to say, is this relevant to samples? If it's not relevant to samples, what is? And how do we modify this text for our community? So we're going to break out into groups, and it's great. We've got enough people online, Val, that I think if it's all right, we can put you guys in a group together. And then I created 3 documents. So if we we can either have 2 groups, one online and one in person, working on the same document, or we can make a copy of it, or we could just do 2 groups in the room. Is that too big though, to break the room into 2 groups? We might want to do 3 anyways. So yeah, Val, we'll pick whatever one that you guys want to work on online. We'll make a copy of that, and so we'll have 3 groups in the room as well. And then we'll just make a copy of one of them so that we've got you guys duplicating one of the groups. So in each group, we'll want you to get together. Hopefully someone has a laptop and can pull the document up. When I teach, I like to tell people in activities, identify a note taker who's gonna be the lead on that, and identify a reporter who's gonna report back. It helps, so then at the end, you're not just like, wait, what? And then during the activity itself, we've got 2 phases. One is a set of questions on the overall document itself, these guidelines, where we just have some conceptual things that as a cluster we've been thinking about and we'd like to get feedback on. Then we actually have, as you can see, there's 3 different sections of the document that we've extracted and turned into a single document that we'd like you to look at. And again, you can either say, this is not relevant at all, we're not even gonna have to look at this. Or you can start editing the text and saying, this is really close, here's just some minor changes. Or it could be something where it's more in depth. But we've got sections on data availability, which is more of a conceptual concept of what it means to be available. how to do peer review, and then finally data availability in a more practical sense. And so again, these are sections of the guidelines that the cluster hasn't yet sort of changed or adjusted. So when you look at these, they're going to be written as if it's for data or software. And so we're looking at, from this, how do you adjust it for samples? And so let's take a look at one of them so you can get a better idea what I'm talking about. So at the top, as I mentioned, there's some questions just as what's relevant in the overall document, and there's a link to it at the top of each section's doc. And then at the bottom is this edited text that is the text to edit, which is in red, and it's just anything that hasn't been touched by the group is in red, so you can, it really stands out from some of these. We've already made some edits, and I'm really sorry, I can't get this to scroll. Sorry, for those online, so you don't get context, the Zoom has sort of taken over the screen, and the scroll bar for any document is off the Zoom screen. But yeah, so just to give you an idea of what you'll be working with. Okay, all right. So at this point, we're gonna break out into groups for 30 minutes. If you wanna self-select into groups, again, I know I saw Mark make a face at the names. They're bad names. We were just trying to figure out like what these sections represented. So we've got data availability conceptual, peer review, and data availability practical. And then you've got the documents with a little bit more instructions in them, and I can hop around to each group to check. And Val, if you want to pick a section that you'd like to work on, I'll just make a copy of it for you guys. Or if you want to just go ahead and pick one and make a copy of it, whatever works easiest for you.
Yeah, I'll just pick one. Maybe we'll do peer review.
Okay. And so then we've got, so at 9:35, we'll come back together as a group and talk about a little bit of what you discovered. And again, if you don't end up making any edits to the text, that's fine, but any reflections you have on it, we'll really take that in so that the cluster can work on that as well. So should I mute this so that we don't end up?
Yeah, that might be fine.
Oh wait, Mark's got a question.
So we're talking explicitly and only about the sample itself, right? Not data derived from the sample.
Right. Well, that's actually one of the questions we have at the top of what should be within scope of these documents. And again,
So, but the data derived from the sample, would that really be different from citing any other kind of digital data?
well, that's why. Sorry, hold on. We've got a mic transfer for those online.
We already have guidelines, Shelley, help me, on how to cite data in a publication, software and models. What we don't have is a reference on how to cite samples. The physical sample and the physical feature the sample comes from. If you want me to split hairs, let's just call it physical samples.
But it is a relevant question because in the group we've talked about, are there any specific needs related to sample derived data that are different from just general data? And we don't think there are, and we think it might be outside of the scope of the document, but we are open to that perspective if you think, well, wait a minute, there are some special needs for sample data that, yeah, we've got some more comments from the audience.
I'm not a physical sample expert. I just want to put that as a caveat. That's why I need you people. But I think what is really special about sample citation is that there's usually a direct link to analysis of the sample. And I think that's very relevant, especially when so you do a chemical analysis of the sample, you do I don't know, the things you do with a sample. The things you people do with a sample, but there's a direct link, and I think that's really relevant and should be considered.
Yeah, and I think that's what we were getting into within the cluster, is that sometimes is that just considered the metadata about the sample, or is that actually some separate derived data? And for those online, we've got some questions in the room, so we'll continue.
I have one suggestion. So, I mean, another thing for us to consider might be how these guidelines work with the data citation guidelines and making sure that there's no gaps in between those two things. Because, yeah, and another thing to maybe consider, and this is probably beyond the scope for today, which is what is the responsibility of the scientists writing the paper to manage as far as like identifiers and so on, and what is the response, and if they're putting stuff in a repository, what's the responsibility of the repository manager or the collection manager?
So-
That's like a harder question.
Well, and in some ways, I feel like that falls under the peer review category of like, as a reviewer, what information should I be getting from both the paper author and what are they getting to reference back to the repository where they got the sample, and have they been given the proper information so that connection can be made?
Yeah, and that last question about data from samples, I'm not sure if it's totally different from earth science samples, but with biological specimens, you often have to process the DNA samples separately. You might even take it from a different specimen. So it's not like there's a provenance necessarily that's shared between the data and the sample, but they are bound together as different collection objects. I don't know if that makes sense.
Yeah, and I think that's where connecting the relationship between subsamples or derived samples and how to represent that might, in some ways, and to get to Mark's point too about the connect, that's one of the things is we need to know, well, where can we just say, is it as easy as saying, we're not covering this here, but see this other document, and making those connection points back to how to deal with data that's already been identified. But we've got another comment in the room.
Oh yeah, this is just a question. I'm also not an expert at all in physical samples. So, but I'm curious, are we sort of primarily concerned here with physical samples that will still be there after analysis, as opposed to something that's completely destroyed during analysis? Because with like, you might break off a chunk for DNA analysis and you totally destroy it in that process.
Yeah, that's, I love that question because people think you still want to know that a sample existed, right? And be able to say that there was a sample and here's information about it, and that even though it was destructive sampling, something existed. So yeah, we are interested in both. And I think Mark had a follow-up point.
We actually did have a raised hand on Zoom.
Oh, do we?
first.
I'm sorry.
Yeah, so feel free to unmute
Yeah,
yourself.
sorry. If the person on Zoom wants to speak. Hi,
Hi.
Diana.
Yeah, sorry. So again, not an expert in this type of work at all, but so it occurs to me because I was trying to think through how your conversation overlaps with all the other data conversations that I've been listening to. So if for instance an ecologist takes a transect of a piece of land and they collect information on it, in theory that transect is the sample, right? And so in some ways, this conversation overlaps with what would be considered metadata in other conversations. And so I don't know if everybody's interested in drawing a really fine line between data and metadata or data sample and data, but I mean I think that there's a significant amount of overlap that the conversation definitely needs to be had between these different communities.
Yeah, and I think that's something that I think Leslie might have touched on earlier before she had the mic, is that idea of the sampled feature as well. And so I feel like with that transect, you're identifying the place and the thing that was studied. And so a sample can sometimes include or has to include sampled feature as part of the conversation. And then Alexis, did you want to say something? Your hand was up and then disappeared.
Yeah, just that sometimes it seems like in the sample space, what's metadata versus data sometimes also depends on the output. So, I think that's a key part of the peer review question here that we'll get into. But, like in what context is this the sampling event, the sampling context, the derived data, or something else different entirely?
Mm-hmm. Yeah, and I do want to say too, these are all great questions. Like, I feel like I don't know if we have an answer yet for some of them, and that's okay, because that's something that we need to then capture and bring back to the community to work on for perhaps in the guidelines. Okay. Are there any other questions or comments before we move into the activity?
Just, yeah, and I guess this is sort of the same question as Diana's, but when does a sample become a first-class object that deserves recognition in its own right? So if I'm digging a snow pit, I take a bunch of samples to measure the density, but then I throw that all away, and all I really care is about the density. I don't care about the sample itself. If I take an ice core, I keep that ice core, and I be very careful about when I destroy the pits to do an analysis. So those are like two opposite extremes, and one's really expensive and one's really cheap. So have you thought about, when does a sample become important and not, I guess, so for the snow pit example, then it's just metadata. The snow pit was in this location. But the ice core, you have that same metadata, but then, you're doing the isotope ratios or whatever. So have you thought about that? When does a sample become a recognizable thing that we care about?
I probably could posit a few things about that. I think because what comes to mind when you say all that is reuse, right?
Okay.
And validating results of something. So your sample in the snow, I can never go back and get that again. That instance doesn't exist anymore. Right. So I want all of your data and information, and I can truth it out or reuse what you've created, but I'll never be able to go back to that spot and get that.
You can't go get another ice core either.
Right, you can't, but at least the core
persists.
exists somewhere in a repository where someone can go back and revisit it and say, okay, now I know this, I want to do more tests, or, oh, I don't believe your test, I want to do my own version of it. And it's part of that scientific process. It's got some value in a different way than maybe the snow that disappeared. But I didn't really want to say that, because then what about destructive sampling? We've now sampled something we had it physically for a while and then destroyed it. Where does that fit on that spectrum?
Well, and I think if you're looking at genomics or proteomics or metabolomics or any of the omics,
you have to make
often you don't care about the specific mouse, you care about the strain of mouse. Mm-hmm. And that mouse is disposable, unfortunately.
And I think that's where in the biodiversity they go to what is the term? Occurrences. It's an occurrence of something and not an actual sample of something, because it's, like you said, it's not the mouse that they care about, it's that it's a type of one of these mice. Alexis, did you want to say something? And then we've got some hands raised in the room too in response.
Yeah, as a recent mouse person, you often do care about the mouse, right? Because you also care about not just the strain, but the marker for where that strain is housed, because there's a difference between a mouse housed at Jack's versus a mouse housed at the NIH. And you also care about the generation of that mouse. So even in the mouse world, the metadata is more comprehensive than just the mouse strain for the sample.
Talking to the mic.
Yep. The mouse itself is not a first-class object. You have all that extra information about the mouse, but I'm asking, I'm not.
Why is it not a first-class object? Oh, Diana or Alexis? Sorry, I saw hands.
Oh, sorry. Yeah, so this is a very, very interesting conversation. What I would actually want to contribute is that I actually think I don't know quite yet how to think about samples. So I'm still listening, but I absolutely think that metadata is a first class object. I think that metadata is something that in the discussion of data, what we'll find out is that metadata is as important as the data itself, because it is the metadata that enables the reuse of the data. And so the richer the metadata, the more use you can get out of whatever it is that you consider to be the primary data. And so maybe samples fall into that, maybe samples don't, but I just wanted to throw in that I'm a big metadata fan.
And we've got a comment in the chat too that says people at Biodiversity Information Standards, TADWIG, and Global Biodiversity Information Facilities, GBIF, are having discussions about this concept of samples, occurrences, events, and specimen. So I feel like it is an unsettled, that's a relief. It's an unsettled issue that we don't have to have an answer for in the room. I guess, Abigail?
Yeah, I guess, so this is Abby Benson. And what I wanted to add is I thought what I heard is the goal here was to get credit to museums and the places that are housing these samples so that they can continue to do it, because they sort of lose credit. So I think in my mind, that's the main focus. So when we go off on these tangents of, the metadata, if it gets destroyed and whatnot, I think what we're really trying to do is get credit to those places that are stewards of these physical things that exist that you can go to and reuse, and make sure that they can be fully credited for doing that, because it's not free. So
Mike? It's Mike.
It's more than credit though, right? It's also the specific reference to the thing.
yeah, I think there's other ways to still, like for an ecological transect, I think there are standards like Darwin Core that exist where you can identify all that information and get people the credit they need. I think what's unique with specimens is that you, rather than a data set or a full collection, you want to have each specimen identified in a paper that's used, so people can say, these are the specimens that were important in research.
And I think there was, the person in the back had his hand up earlier. Did you want to share something? The hoodie? No? Okay. And then, Leslie, and then, I'm sorry, the guy in the next row behind you as well.
Just go back quickly. This argument also, I seem to remember, happened with data, where it was you gave an identifier to a data set that was cited in a publication, and now we use data site DIs for all sorts of things that aren't necessarily cited. In Australia, and I think Kirsten was doing it in the US as well, we were actually assigning the IGSNs in the field. Because if you were there and you collected something and you brought it back, we didn't have to then wait until, because if you wait till it's at the curation stage when you've done something, then any sample splits off that are impossible. And so that's why we were giving people the option of it not being a tool for museum creators, but for agencies. I was at Geoscience Australia at the time. So when I went out in the field, the minute I picked up that sample, I gave it a globally unique identifier. And then, as I said, I brought the damn thing back, and God knows who did what with sample splits and everything went on that sample. But you could always cite back to the museum, and I presume it'd be the original mouse. Someone still has that mouse or something there, and people can go back with a bit of luck and get more material from it, or can trace what somebody else has done in the literature. Getting fit in this session, aren't you?
Was there another comment or question earlier? Somebody? Okay. And then, yeah.
So this, again, might be sort of a naive question, but so how would these samples, the physical samples, differ from other physical objects that are handled by libraries?
Well, I say, in my mind, that's what got me into this, right? I didn't think there was a difference. I was managing a repository and I was bringing in library science concepts, and everyone was like, this is amazing. And it's like, this is libraries. So it like, yeah, I don't see, except for if we go to, I'm gonna geek a bit, Ferber, when you think about functional requirements for bibliographic references, is this concept of like you have a manifestation of a work, like there's different levels of, am I talking about the concept of the work? Like, am I talking about every version of the copy of the play Hamlet, or am I talking about a specific version of it somewhere? And for samples, you end up, not always though, saying I want the specific version of the sample that is someplace, but theoretically in some fields, I say theoretically because I don't work in those ones, but I think it's true, is you might want just, I want a specimen of this type, and then it doesn't matter which one it is or where you're getting it from. But I think because of, as we see in some of these conversations, people want the one that somebody did something to in order to look at that same thing again for some purpose. And so it gets to be a little bit more heavy on that end versus libraries, where you have many copies of the same publication and it can be copied and distributed, but you can't necessarily copy and distribute a sample. Val?
Oh, yeah, I guess I was just kind of gonna give the example just from, I work in a physical sample repository of, you don't like rip a page out of a book and then hand it to somebody and it may not come back to you. So just definitely a ton of similarities, but.
And then Diana?
Yeah, so I wanted to, so I actually really like your concept of Hamlet, and I wanted to throw in, you know, so you were mentioning that the AGU has done a lot with software documentation. Another thing that you might want to consider with regard to this, with regard to samples, is if you care about a particular version of Hamlet versus Hamlet, software deals with very much the same issue with you know, whether one person has a branch of something from GitHub or they have the authoritative, you know, whatever piece of software model. And so that may be something to consider in this discussion of samples.
And I think that's why we use the data software guidelines as a base, because there already is some conception of like, it's not what version of it is it, and where is it coming from, and a little bit more of that provenance behind it as well. So it was a good base. It just then there tends to be differences. Like you can't necessarily treat the sample the same as you would data. Any other questions or comments in the room? Just thinking too about time now. We we would have had 10 more minutes in the breakouts, and I'm trying to figure out mentally, we've got 35, this session goes till 10, right? Okay, so we have 35 minutes. This has been really great conversation though, so that's why I didn't want to say like, no, let's stop this and go into the breakout groups, but I guess we can get a little bit of a feel around the room. Do we want to continue to have some of these conversations, or do we want to now try to look at these documents with a little bit less time? Just sort of get an idea of your impressions of them. Again, you can look at just the guidelines themselves, which in each break activity are linked, and look at some of these questions about it, or you can actually start jumping into any of the text. So if people are okay with doing that, we'll maybe do that for, let me do the math, for like 15 or 20 minutes, and so that we can save again the time for coming back and reflection, because I think we've already covered maybe some of what we would have done in the breakout reporting, but this way we will still save some time for that. So then it sounds like online, you guys are going to look at the peer review document, and Val's already made a copy. And then in the room, we can divide up into the 3 different documents where the data availability on a conceptual level, peer review, and data availability on a practical level. So if anyone's got like I want to do that one, maybe we'll break up into 3 groups in the room. And so I'm going to mute the Zoom both so that we don't talk into you guys and that we don't hear you guys talk, and then we'll come back in about, let's say, 15 minutes. Okay.
All right, virtual participants, everybody, how are you all doing? Are we in the
Hello.
document? Hello, Diana.
Morning.
Yes, good morning. Well, unfortunately, I've been trying to my picture cannot appear because I guess you guys have turned it off. So anyway, so I apologize that I'm not there in picture wise.
That's okay, it happens. It's really early here, so I definitely contemplated just not showing my face.
Yeah, I'm getting the same message.
How strange.
Yeah.
The host disabled my video.
Yeah.
But it's not a personal thing.
Yeah, so it's not personal.
Oh, maybe this is because they switched the host when our ESIP fellow supporter came in.
That's right. My office is really boring. No one wants to.
Okay, so does anyone need the link for our slides just yet, or for our breakout, or is everybody good to go?
I've got the document that you sent.
Yeah.
Great, and you can get in there because I definitely just copied it.
Yes.
Haphazardly in it. Great. Okay. Also great to meet you guys.
Yeah, definitely.
Okay, let's see. So I have a timer going on my phone for 15 minutes.
So can I ask really quickly if we
You
can
bet.
introduce ourselves so that, because I can't see and I'm totally new to ESIP, so
Let's do that. Yes.
just really quickly around the room. I'm
Yeah.
Diana Ma. I'm from Hippocampus Analytics. I am a former neuroscientist, now technologist interested in data citation.
Great. I guess I should go. So I am one of the co-chairs of this ESIP cluster. My name is Val Stanley. I manage a collection of deep sea sediment samples from the Southern Ocean here at Oregon State University.
Marcella Redden with the Geological Survey of Alabama, and along with being a geologist and researcher, I'm the administrator for our core repository.
Oh, Marcella, I think we've met here before.
Yeah. Yep. I don't get to make the cluster meetings. I have something else that's at the exact same time most months.
Even since we've moved it?
Yeah, actually, you moved it to the same Monday that we keep having this meeting, but that project's only 2 years long, so it's cool.
Ah, geez.
It might get moved again in the next semester. We'll see what everyone else's schedules is. So.
I'm Alexis. I was a CIP fellow for 2 years with the Data Stewardship Committee, so that's sort of my inroad to ESIP. But I am now a doctoral student at the Jackson Lab up in Maine, and we handle all the mice that are used in mouse research, so hence the mouse piece. But I'm also the data and information researcher with the Mohawk Preserve, which is a small institution that has a pretty large catalog of historical specimens and objects that they have on site that I help manage, which is my interest in this cluster area.
Excellent. All right, great. From what I can tell. All right. Anybody else want to do an intro real quick? I can see a couple more folks online. All right, great. So if it's the four of us, which is wonderful. And we have Diana, we have some new folks here. So, and especially since we have 10 minutes, do you guys want to dive into maybe just 5 minutes of us just looking at this and sort of commenting in here?
While I'm reading through this, maybe somebody can talk about the issues that people care about with regard to peer review of samples. I have some sense of mouse samples because I did work on mice. But in earth samples, what is reviewed? I mean, you care that the sample is reflective of the data.
So that's a great question. And from my perspective, it's that there has never been a standard for this. And that's why this is a particularly interesting section. Because, I mean, presently, it could be in the acknowledgements that someone says, samples are available, email me. They may or may not be in a repository in an official space. Someone may not have access to that.
Right.
In some cases, there may be a very
limited
robust,
sample,
like resource that people can go find something and you can say contact this curator.
Yeah, like moon dust.
Mm-hmm.
Sure. Moon dust. I mean, it's like you have to go to NASA to get some, right, or something like that. But
yes, but they may have a good process for that, and it's
right.
They can go there.
Yes, yeah, exactly. But so how do you generalize that process versus something like a sample of temperature and salinity from the ocean? Like how that, I mean, would you even consider that to be a sample if you take up a patch of water so that you can measure its salinity and temperature? So anyway, I again, I'm fascinated by this topic.
Yeah, totally.
But I don't know anything about it, so.
water that you collected for salinity and then discarded would be similar to what Mark was, I think it was Mark in the room, saying about collecting a snow sample but then discarding it rather than a sample that's preserved.
Yes.
That's my intuition on it.
Yeah,
For sure.
I think that's absolutely true. And so in which case is that considered then metadata, and how do you draw a line, or do you need to draw a line?
Yeah, and I also am thinking about this from the repository perspective of, say we have
Exactly.
a sample and it's exhausted, we still maintain the record for it existing.
Right.
Because that sample did exist.
Yes.
And data were generated from it.
Right.
So it's kind of, you just sort of change the availability or the condition of the sample to depleted.
So maybe that needs to be in the documentation, is that it was discarded, or that it was ephemeral, or.
Yeah, ephemeral was a word that I put in the notes. Sorry, I've been secretly taking notes on the whole
Yeah, good.
conversation so far. Yeah, ephemeral was something that I was thinking of, like things that are known from the get-go. You know, the sample is going to be, it's not going to exist forever.
Right.
And another, yeah, I know, thinking about that is, there's a lot.
Yeah, so I guess, you know, I mean, I'm trying to think of it from the metadata point of view, and I'm curious whether samples, whether it is ever inappropriate to consider a sample part of the metadata. And because again, this is completely my perspective, but I consider metadata to be a first-class citizen, and so for me, it's not downgrading the sample by any stretch of for the repository people. It's not downgrading the sample. And I'm just saying that it's treated in a way that you know, you can write it's ephemeral and it's not a sin to throw it away, because people who are data people consider it to be a sin to throw away data, right? People who are software people don't consider it to be a sin to upgrade software. So you know that it's I think that this is a fascinating discussion.
Yeah, I just made a note that there needs to be a way to communicate that the sample is not and cannot be made available.
Yeah, that's a really good point.
Yeah.
One question I have is just for framing this. This is for peer review of articles or objects that use sample data rather than peer review of samples themselves.
Great question. Okay, so the entire document itself kind of has three chunks to it, like that we've considered like three kind of separate things to consider about citing samples. So there's citing the sample itself and its physical object of where it is, as well as the metadata about the sample. So, you know, has this person given it an IGSN? And like, where does that sample metadata live? And then also the data about the samples is a third piece. So if you've, you know, measured salinity of your water sample, that's separate from the sample itself. So that's kind of the three aspects that we've considered so far.
Great, thank you.
Yeah, but I guess, and so this peer review idea is, you know, what would you say to an author who's like, okay, so there's a requirement for me to cite my samples, how do I do it? And so if there's a peer reviewer, or, you know, it's going through that, what should that person consider? So, I mean, you know.
Okay, so
Go ahead.
I hate to do. I'm trying to throw in all sorts of different monkey wrenches to see where the system breaks. What I'm thinking about is, so I started life as a marine biologist. We would take fish as samples, and we cared about the state of sexual maturation, whether or not they were ready to breed or not. But then we would also take a number of them and put them into alcohol for preservation for later analysis. And so in this question of ultimately the goal for the scientist is attribution. The goal for a repository person is you need to track this thing. I'm just thinking about many things. It is no longer useful for measuring certain things if you go back to it 50 years later, but it is perhaps more useful for measuring other things, like if you cared about the isotope 50 years ago, the isotope levels 50 years ago. And from the repository perspective, from the repository perspective, I suspect you still treat it as the same thing, but from the researcher's point of view, it's a very different, it has very different uses. A sample as versus digital data has very different uses 50 years later, right? And so which matters more? Do they matter? Do they both matter? And how do you reflect that in the fact that it's a sample for peer review? To give credit to the sampler or the repository museum.
Interesting. I think this is based on my experience popping in on the research artifact citation cluster occasionally. This sounds a lot like some of the things they worked on. Have you, so they had like a chart and they were like mapping out like who all are the entities that need to be cited. And like how do they interact with the data? And so hopefully they'll be like sussing this out with samples as well, or we will be at some point.
Yeah. No, I mean, I think that is absolutely relevant to know if I am the scientist 50 years from now that wants to make use of a sample. I not only care about where it was sampled, what it was, but I care about how it's been maintained, the fact that it is 50 years old, how to use it properly and to not misinterpret what it is in my analysis. So anyway, this is a fascinating conversation. I love this conversation.
Yeah. Okay. So we're down to the final minute here, and I see Sarah coming up on us. So are you guys okay during the report out if I just kind of let everyone know that we got up to speed on it, and we've been just kind of grappling with the idea of how peer review, citation, and ephemeral samples all intersect and how to move forward with telling people how to work with that?
I'm great with it.
Great. Yeah, we'll do that. Thanks, everyone. This has been fun.
All right,
That's
everybody in the room, we're going to regroup. They don't want to.
All right, we are going to regroup. Helping Sarah out here.
I know, we're trying so hard. It's so exciting that people are so passionate about this stuff that they don't want to stop talking about it individually and talk about it as a group.
I want to know what they're saying.
All right, for those in the room, the people on the line want to know what you're talking about. So we're gonna like pause the group discussions and allow you to report back so that you can share these exciting things you've been talking about with the people online. All right, I think they finally got the hint.
Thanks, everyone.
All right, so who wants to go first and share what you guys have been, a few key points about what you guys have been chatting about? You were just so passionate all a second ago. All right, Val, why don't you guys online share what you guys were talking about first, and then we'll see how it connects with what people in the room have been working on.
Yeah, sure. And I'm also very curious to see how much overlap there's been, because we also had a very interesting discussion. So since we had folks in our little group kind of with different levels of engagement with this project, we kind of talked a little bit about the project in general and like kind of the breakdown of this, the structure that we've been working with, and discussion about how we've kind of broken out the topic of physical samples between the object itself, its metadata, and related data. And then when we kind of took that and went towards peer review and considerations for what we need to talk about there, we basically got wrapped up in a discussion about, you know, citation guidelines are very relevant to peer review guidelines. And we also really need to consider ephemeral samples and what to do about that, because a repository may have a very clear way to manage that over long periods of time, but that's different from the researcher perspective. So what is I don't want to say this went all the way back to like, well, what constitutes a sample? But yeah, just the grappling with the transient samples and how those may be different from samples that have been consumed over time slowly.
Yeah, I see lots of nodding in the room. I think everyone agrees that's a really fascinating and interesting point.
Yeah, and so basically just, you know, think about that and what information is needed so that we can then kind of funnel that into what needs to be considered for peer review on those kinds of things.
Cool. All right, does one of the other groups want to go next? Andrea?
Yeah, we were all nodding and laughing because we had probably the exact same conversation, which led to, what is a sample? What are we citing?
That's amazing.
And also thinking about like, basically, what are the use cases for, why are we doing the citation? What are the use cases for needing to cite something? So we do think that the main reasons for citation is to provide credit and attribution to the repositories and the people doing that maintenance work, that curatorial work, but also for transparency. And I do think that the transparency argument is important to get scientists on board who might be less altruistic towards the people that are maintaining their data. But like, so I think those are the two main samples or cases. And we also started talking about like that same sort of issue of the ephemeral sample. And so I tried to think of that as like if your sample was destroyed in analysis, if your sample wasn't destroyed, but it's not in a formal repository, and if your sample is in a formal repository, as like 3 cases for like how to cite and what information to cite. And
oh, good. I hope you put this in the notes.
I
right?
did.
Okay,
Yeah.
good.
But like we are not totally, like there's still some points of contention within that. I think that there's, Mark was talking about maybe suggesting guidelines for repositories. And I think we need to know more about like, what are the relationships with different repositories and infrastructure like CSAR and IGSNs? Like how hard would it be to recommend that repositories use IGSNs or something like that, which is, I know, part of the bigger iSamples project. So that might be context that could be helpful. So Abby, did you want to add anything, or anyone else?
I'll just say too, I think that's useful as far as what supplement documents need to go along with these guidelines. And that was one of our bigger questions too. So that's helpful.
Well, so we also discussed that while there are those 3 use cases, maybe narrowing to one to focus on first instead of trying to tackle them all 3 at once. And some of us had said, well, go with the route of, you know, samples in a repository, like start there, but others felt differently. So like that needs to be a discussion on what to tackle first, but I think there's agreement that start with one.
Yeah, agree.
Could you repeat that into the mic for those online?
That could be a good way to split up the group according to these different use cases.
Thank you, yeah.
But then also I think, so we're starting with the AGU guidelines, but the AGU guidelines for data and software are built upon the ESIP guidelines. So the AGU guidelines assume that there is enough information for the author to cite the data. And we're not at that state yet.
Yeah.
And so it seems to me that whichever of these use cases you pursue, if we do the ones with repositories, it's probably, from my perspective, since I'm clueless in this area, we need to understand the cultures of the different types of repositories. I think museums that handle life science collections are very different from repositories that hold rocks and ice cores and things like that. And then when you start looking at, when you start doing genetic analysis of different things, that's yet another sort of creature. So I think there's also not only these different use cases, but the different cultures of different repositories.
I hope you captured that in the notes too, because I think that's a really good point. Okay, yeah. Because with some of the earlier parts of the guidelines, we've been looking at when you make a recommendation of where should you put things, and I think that culture is important for that too.
I'll say one last thing before I surrender. I know it's annoying to do this, but defining what our group means by sample would be a good starting point to keep us from continually redefining it every time we have a meeting.
All right, and then does someone from the last group want to present? Because I will call in Denise if no one else volunteers. Sorry. That's fine, that's fine.
Okay, so we talked a lot about, again, going back to the purpose of citation and how far do you kind of go down that wormhole of what you're citing, data set versus sample collection. There was a lot of discussion about what are you supposed to be citing every individual sample within that collection or just the collection DOI, and where is that? But then if your analysis is of a subset of that collection, where are you recording which samples you're getting that data for? And that's where we kind of got into that conversation about supplemental data not being a good option, but then we don't really have a good option. And then again, it's perspective, like a lot of the conversation was coming from a researcher perspective, but me coming from a repository, I'm thinking of, well, I want all these other connections because it helps me do my job to help our users and protect our collections. So kind of finding common ground in that as well.
I think capturing those perspectives really goes to what Mark was saying with the culture too. If we can say, like, this is why we're asking you to do some of these things because this is what it does within our community, there's a great value in that. Is there any other comments from that group? I think just because I think we're running to time, we'll probably just, okay. All right, so originally we were hoping to then talk about next steps for this, but I think as we're running out of time, these are just things to think about, that if you have any suggestions about this, I think what we're still going, it sounds like everyone likes the document, likes the idea of working on the document, and that we're gonna continue moving forward on it. We do have some suggestions of potential supplements or connections to this. And we already have earlier talked about some different places where we might take the guidelines. So I think we sort of touched on some of these. But the main thing here is, and I'm gonna skip some of this stuff that we had planned, was just to say that you should sign up for our listserv to stay engaged in the community and in the conversations if you haven't already, and that we have monthly telecons where we're going to continue these conversations. Right now, we're scheduled to have the next meeting on August 15th at 4 p.m. Eastern. But we've discovered that even though we just picked this new time, it turns out it's not a very good time for anybody. So this is not a hard and fast time. If I can get my act together before August 15th, we'll put a poll out to determine a better time. Otherwise, we'll probably just keep this for now and pick a new time for September. But we strongly encourage you guys to join the listserv and the lease to stay engaged with these conversations, even tangentially as we move forward with things if you're not able to join the monthly calls. And then if anyone has any ideas, I want to throw them into the breakout documents because I don't want to keep you guys as we're starting to move into the break. ESIP does a really wonderful thing of having, after the plenaries, they'll have these forget what they're called, but these sessions where it's like 1 minute for every cluster or session to like report out the outcomes of what happened in their session. And I really like that as a concept, and I like to take part in that. So if anyone feels strongly about something that they'd want to share as a takeaway, to say, if you're going to talk to the rest of the ESIP community about what we did here in this session, either if you want to quickly shout that out now, or add it to the notes, and then we'll probably give one of these presentations to share that back. But thank you all. Thank you all online it sounds like you were wonderfully engaged, and thank you all in the room as well for joining us today. And we're only 5 minutes over, so not bad.